from the Central Institute of Petrochemical Engineering and Technology, Guwahati, India. She's gonna be deliver the talk on polymer matrix-based core shell nanoparticles for medical technology. So before that, we like to have the scientific accomplishment on it. So she had done a PhD in the domain of nanoelectronics of next generation biomedical and wireless communication devices. And also she is the awardee of scientist, women scientist, a program from the government of Department of Science and Technology, Government of India. And also she expertise in microwave processing, and also she worked in German Research Foundation and also the Indian Department of Science and Technology. With this, she has 5 years of research and teaching experiences of teaching postgraduate and the PhD level at Guwahati University, Assam. And she also have a different domains of research with microwave frequency of medical application, core shell nanostructures, and have published one book and also many book chapters, and delivered talk at various international events on nanophotonics. And she's a very frequent coordinator in AICTC, ATAL, FDP, and also DST FDP programs, and core shell nanocomposites of the biomedical imaging or her thrust area of her research. And she also got recognized as an innovator for a worldwide academic industry network and the focus of communication and medical technology related stuff. So this is the where the point of ATAL program is a point of contact of both Dr. Jyotika Devi with our research group. So with this, I like to welcome Dr. Jyotika to deliver talk on polymer matrix-based core shell nanoparticles for medical technology. Now over to you, ma'am.
Thank you very much, Dr. Mubarak, for these encouraging words. And I wish all the women participants a very happy Women's Day, though it was on yesterday, but still the theme is also very vibrant one. So just I'm sharing. Is it visible?
Yes, yes.
Okay. So thank you very much, and it is a feeling that in this time I got the opportunity to join the International E-Conclave, and with special reference to biotechnology, since all the fields are interdisciplinary, so it is quite nowadays it is quite normal that people from physics or people from chemistry or from an engineering can work on bio, and bio people can work in other domains, because everything is interrelated. So here I am primarily focusing on the core shell nanostructures for medical applications and how the polymer matrix can be used. So as we all know that nanotechnology is working on measuring, manipulating, and controlling at the atomic level, and its length is approximately 1 to 100 nanometer. So in order to understand the how the material will work as a device, how the system will work, so it is quite a very special thing, because the properties are the explicitly depending parts here for the nano. And how small is nano? Those who are from nano domain, they have the idea what is 1 nanometer, but as a common people, how small is nano? So a piece of paper is also 1 lakh nanometer thick. A human hair is 80,000 nanometer thick. So comparison, if we compare with a DNA is 2.5 nanometer of diameter, and a gold, the atom is 0.3 nanometer of diameter. That means how small the nano is. So it's the 80,000 nanometer thick a human hair. That means 1 nanometer is 80,000 times smaller than the human hair. So it's quite a practical thing that if we want to know the properties of a nanomaterial, we need some sophisticated device. And in our human cells also, just like virus, then bacteria, these things are falls into somewhere around the nano region. So if we are finding just like antibodies, so this nanomaterials plays a very important role in diagnostic of the diseases as well as the treatment of the diseases. That's why nanobiotechnology has popularized in such a way, and its growth is expanding day by day. And as nano is there, we know that for nano, size is always important. So the main specialty of a nanomaterial is that size does matter, and surface area is inversely proportional to the size of the particle. Here, if we see that in the microparticles, the volume matters, but surface area is not. But in nanoparticles, since the size is very smaller, so more number of particles can be accommodated. So at the interface, at the surface, its behavior is much more important. So that's why the point of interfacing for nanoparticles are playing a very important role in the disease treatment also. And another part is this nanomaterial size is in 10 to the power minus 9 meter is 1 nanometer. So this particle shows wave-like behavior also. And here when it is wave-like behavior, the classical mechanics, the normal mechanics which works for the other macro particles or macro size objects, that doesn't works. And for this, quantum mechanics plays a very important role. So as those who are from physics, chemistry or science background, they know that this wave-like transport means that the particle size is comparable to the de Broglie wavelength. That means, but those who are not from science background, for them also this is a common thing, that means they are very smallest type of particles. And since it is in Armstrong, just like I have said, 1 Armstrong is 10 to the power minus 10. So 10 to the power minus 9 nanometer. That means those works at the source maximum, though they have the particular type of nature, but they show the wave-like behavior. And when they are wave, that means at that time their particular nature is not dominant one. The wave behavior, they behave like a wave, they behave like a signal, that comes into play. And the nature of electron in nanostructure is different from microstructure, since it primarily depends on the size. And these are some common things where we are using a same material. We are using a same material, but we have changes the sizes. That means when we prepare the nanoparticles, if we arrange their concentration, or if we synthesize them in such a way that the size of the nanoparticles get different. Suppose for a 10 nanometer nanoparticles, what will happen? The size will be different, and for a 500 nanometer nanoparticles, the size will be different. In that way, suppose for a 5, in this case also, when the size is different, then what will happen? Depending on the size, the nanoparticles application will also be different. Just like this is a sample of cadmium selenium. So for this cadmium selenium also, this is a 2 nanometer. Just a minute, please. So this is a 2 nanometer particle size. So this is a 2 nanometer particle size. 2 nanometer particle size. So when it is 2 nanometer, cadmium selenium is 2 nanometer. At this 2 nanometer, this color is different. And for this 8 nanometer, this color is different. That means for the same material, we are having different colors, just for the different size. And this size, how we are making this size? We are making this size in by synthesizing it in a different way. So what are the main factors which depends on what these nanoparticles are depending? Those nanoparticles are depending on the size, shape, composition, surface charge, surface corona, and aggregation. So these are the main properties on what for the biomedical applications also, these nanomaterials are the potential candidate. And for these nanomaterials, these physical properties are very important. And if we say the size is the most important thing, because when this size is so smaller, that's why it shows wave-like behavior. And since it is showing the wave-like behavior, so the mechanics which is utilized for this is different. It is quantum mechanics, and since it is quantum mechanics, so de Broglie's wavelength and this Schrodinger's equation comes into play. Another important point is shape. So how shape can be differed when how the spherical nanoparticle shows its behavior, and how the triangular shape shows behavior, and if we combine it in a heterostructure, it will be different. And another point is composition. What is composition? Suppose we have one gold nanoparticle. This gold nanoparticle we are mixing with a polymer material, or we are mixing with a dielectric material, or we are mixing with a two dielectric material, and where one is nano and one is micro. So we can make in this way a functionally graded material. Means these are not normal material, these are engineered. So this type of composition also we can make, and these compositions also we can use for medical application. And definitely surface charge, since in a nanomaterial, if we are using a heterostructure, then two other type of material will be there. So their polarization will play a very important role. And this polarization will affect at the interface also. So in an aggregation, just like we know everyone, uniformity and agglomeration are two different type of things. And when agglomeration is there, that will be different. And how the nanotechnology impact the global economy? The nanotechnology is just like the different other type of things. Nanotechnology is a rapidly growing sector, and nanotechnology plays a very important role in the global economy. And just like nanomaterials, nanotubes, nano devices, everything plays a very important role in the growth of the nano sector. And the global nanotechnology market is expected to reach $33.7 billion by 2030 from $1.76 billion in 2020. And if we speak about the materials, just like we know, nanomaterial has also different types, just like quantum dot is there, quantum well is there, quantum wire is there, then carbon nanotubes are there, nanofibers are there, quantum dots are there, core cell nanoparticles are there. These core cell nanoparticles we are making from two components. That means we are using two single component material to make this core. That means we are coming to them. And all these materials are extensively explored in the recent years as a potential building blocks for the nanoscale devices. And we know that polymer nanocomposites, why it is so special and why it is practically and experimentally used for the medical domain, because polymer nanocomposites can consist of it may be monomer, it may be polymer, and those can be what we can do. These nanomaterials, if we consider it as some fillers, then if we consider the polymer material as a host matrix, then we can disperse the nanofillers into the polymer matrix. And what will happen? This polymer may be in micro size, but the nanomaterials will be in the nanometer range. So combination of both these will make its properties very important, and that is the main advantage of making the composites. And there may be of different shape, but at least one dimension must be in the range of 1 to 50 nanometers. And as I have said, nanotechnology is a rapidly growing technology, and there are many more industries which are exclusively working in this, just like Advanced Diamond Technologies, Advanced Nano Products Co. Limited, Ultra Nanotechnologies, Bracker Corporation, Spins Nanomaterials Limited, etc. Many more companies all over the world are investing and focusing and making their markets. And if we Google, then also you'll get that there are different companies who are manufacturing or who are synthesizing the nanomaterials. And when they are synthesizing the nanomaterials, that means that is their market. They are taking the market by synthesizing the nanomaterials as per the requirement of the customer. And this requirement of customers may be like, it may be of 2 nanometer, it may be of 10 nanometer, it may be of 50 nanometer, means according to the requirement of the application. Suppose someone wants to use for some photovoltaic application, someone wants to use it for solar cell, then their size of the nanoparticles will be different. And suppose someone wants to use it for targeted drug delivery, then its particle size will be different. And normally for our medical applications, the size should not be so much lesser also, or the size should not be very bigger also. There is a particular size band on what the nanoparticles will work for biomedical applications. And these are the applications of nanotechnology, as it is almost all the sectors applications we found, just like agriculture and food, defense and security, energy storage, materials, optical communications, electronics, biomedical, biotechnology, cosmetics and paintings, means everywhere in all the fields, this plays a very important role. And everywhere we are getting the applications. If you go to the defense, they are also just like clocking, and for different type of navigations, these nanomaterials are used. And in electronics also, biomedicals also, cosmetic and paints, just like the sunscreen, lotion, and even we go through the digital markets, just like nano washing machine, then in light and then in display also nanotechnology is used. So in the market also there are many more products where nanotechnology is used, and there are many more products which are in the nano in the research lab. And for medical technology also, just like in imaging, we can use these things. And this nanoelectronics is just like not all the eras or all the decades are governed by a particular things. Just like textile, in earlier 1800, textile governed the market. Then railway comes from 1853 to 1913. Railway plays the important role. Then 1913 to 1969, automobile. Then coming the computer. So computer from 1909 to 2025, and after 20, now just like the distributed intelligence, now artificial intelligence system is playing the role, and it is the prediction, and this prediction is done earlier, but as per the prediction, it is found that everything is working in this way. And now if we see, data science plays a very important role, because after computer govern the sector up till it is expected that till 2025 computer will govern the globe. And after that, based on this, the intelligence system, artificial intelligence, everything will come. And up from 2025 to 2081, it is predicted that nanotechnology will play the important role, and it will govern the revolution, industrial revolution that will govern the globe, that will be nanotechnology as expected. And now coming to the point, if nanomaterials we are using for, suppose, biomedical application or medical application, then many more things will come. First, material will come. So what material we have to choose, and how we predict the performance of a nanomaterial, what are the properties we have to study, the inherent properties or the external properties, everything plays a very important role. And biomedical, how can we predict whether the nanomaterial will remain stable in the human body or not, and how environment will behave. Just like suppose we have a tumor inside, and we want to target suppose some material as a medicine. Then when we are targeting the medicine, suppose that medicine is of different types, so how it will behave, whether it will affect the other cells or not, everything should be studied. So each field pursues research relating to the discipline specific question, but it is a common kernel of questions and answer that these advancements of each individual research field is actually correlated, because material science, in the material science, the scientists are making new materials. They are speaking that yes, this can be used for medical application. Then again, the other people are studying whether toxic, suppose like chemistry people are studying whether toxic or non-toxic. Then when it is coming to the biotechnology, that in the medical domain, people are whether it can be implanted or not. So everything is correlated to one another. And these nanomaterial system parameters should we measure, and how to predict their behaviors, uniformity, transportness, and how it can be used. So these are some of the applications. Just like these are the applications of polymers, how polymers are used in biomedical, just like prosthetic materials, dental materials, implants, medical disposable, dressing, tissue engineering, drug delivery. As I have said, drug delivery. In this drug delivery also, what we can do with polymer materials, we can use nanomaterials. So these are also some key medical utensils or things we used, and these materials we can use for, suppose like this is a saline bag, here also we are using polymer. These are the aids or components which we use for, and here also polymers are used. So these polymers are used in tissue engineering, these polymers are used in implant, polymers in antigens, modified genetic delivery, and polymers are adjuvant. That means almost all the serious or all the areas where particular attention is required. Just like suppose we have in our on our hand cut it by knife. So that is a common normal problem that suppose we sometimes we get owned by something that can be treated because it is visible to us externally. We can view it and we can treat it, just like we can put that on. But suppose something is happened inside our body, just like a cell is distorted and we have to treat it, then this will not work. Then we need some minimized or directive or compact type of things which can be used for those applications. And polymer normally used, and polymer has ample applications for biotechnology, because polymer is transparent and polymer is highly dispersive, and it is uniformly dispersed. This uniformity and transparent behavior or polymer makes it a very effective candidates for the biomedical applications. And these are some, just like the dopamine based nanomaterials also, these are used. So dopamine, we all know, when dopamine level is upper or down, then people get affected from the, when it feel low. So these things also correlated with the nanomaterial. That means drug delivery, photothermal therapy, theranostic, antimicrobial, tissue engineering, bioimaging, biosensing, everything can be in all the sectors. Nanoparticles have, even the depression management, there also people are working on how the nanoparticles can be used or not. So these are some of the polymeric nanoparticles. These polymeric nanoparticles, just like carbon nanoparticles is there, magnetic nanoparticles are there. So these nanoparticles has different applications. Suppose like iron oxide. This iron oxide, if it is suppose Fe3O4, that one we can use for memory application. Just like our brain is the main functionality of our human body. That is the actually main organ which can signal our all the other parts of the body. So this iron oxide and an assembly of iron oxide nanoparticle can be used for storing and restoring data. Just like it can mimic the activities. The researchers are predicting that these magnetic nanoparticles can mimic the work activities of the brain. So just like this gold and silver, this gold and silver can be used for functionalized disease treatment. Just like suppose we have to target or we have to give some disease to a particular cell, then what the doctors also do that. Suppose let's peptide enzymes, those things also, because this silver is not making hazard or any harm to our human body. So this silver is basically used in the silver nanoparticles. The drugs are functionalized, then it is injected to the body, and then it is disposed there. Just like graphene also, carbon nanotubes also. Then alloy nanoparticles means we can dock many other nanoparticles to these magnetic nanoparticles, and we can make its different applications. So just like just before a few minutes I have said, suppose earlier we know that resistor, capacitor, and inductors are used. But now what it has found in 1971, that memory store, that means memory resistor. Just a few minutes ago I have said that this iron oxide nanoparticles can be used for memory application. That means resistance is static. Earlier we know that resistance is static. But when it is memory store, that means memory resistor. That means resistance is not static. It is dynamic. That means according to the variation of space and time, the resistor is different. So this activity, just like our brain has also on state and off state. We can store some memory and we erase some memories. Just like we remember someone's name for very long year, but we forget a particular thing for a short duration. That means on and off states are there, and that means charge trapping and detrapping is there, and this mechanism, this memory strip mechanism, can be used in point of care disease diagnostic also. That means if we have a assembly of nanoparticles, and if these nanoparticles we can make a device, just like two electrodes are there. If we are applying voltage, then current, we can record the current, and since it is nano material, so we can use the source meter. And then what will happen at every second of time, since space is related and time is related, at different time, at different space, we can record the current. That means it can, suppose someone is having a disease, suppose we have normal blood. So normal blood, the VI characteristics curve will be different, and if we have some blood, suppose, which is having disease, for that this VI characteristics will be different. So depending on this voltage gap or current gap, we can say that yes, this people, this is sick, and he or she is infected by some diseases. So these are some materials. As time is very short, so I am just speaking on some few important things, how it works. So suppose we have a nanomaterial. So if that nanomaterial has permittivity, the dielectric constant greater than 0, then it can work as a capacitor. If the nanomaterial which is having a dielectric material which is having the permittivity less than 0, then it will behave as an inductor. And if this nanomaterial is there where this real imaginary part is not equal to 0, that means real part is greater than 0, it is capacitor. Real part is less than 0, it is inductor. And if its imaginary part is not equal to 0, that means it will behave as a resistor. That means the resistance, capacitance, and inductor. Earlier we used some lump elements. So that lump elements are big elements. So they are components. So when we are using nanomaterials, what will happen? The small nanomaterials whose inherent property, dielectric property, is greater than 0, they will behave as capacitor. The dielectric properties is less than 0, that nanomaterial is behave as inductor, and the nanomaterial which is having imaginary part not equal to 0, that will behave as a resistor. So normally this, suppose if we consider a dielectric material, then what will happen? Suppose cadmium sulfide, zinc sulfide, like that, they are having real part greater than 0, so they will behave as capacitor. If we say about silver nanoparticles or gold nanoparticles, they have the real part less than 0. They are metamaterials, so they have negative refractive index, so their real part is less than 0, so they can be used as an inductor. And normally almost all the material, whether they have real part greater than 0 or real part less than 0, normally imaginary part is not equal to 0, excluding air. So we can use any kind of material as a resistor. And as the step I had said, suppose this is a shape nanostructure. So in this nanostructure we have shape. So in one case, what happened? The real part of the dielectric part is less than 0, and for other one, real part is greater than 0. That means the same compact structure can work, give us the L and C together, because real part greater than 0 is capacitor and real part less than 0, that will be inductor. So in this way, we can arrange the circuits also. That means that nanomaterials of can give us a complete circuit. So this is the core cell structure, and in this core cell structure, this is the core, this is the cell. And normally what happens, this core is of different material, so cell is of different material, and this can be used for bioimaging, gene therapy, biosensor, drug delivery. So these are the main important domain of our medical technology, and in almost all this core cell nanostructure can work. So this is a core, this is cell, and here suppose this is a matrix. So a medium is there. Suppose this medium is polymer, and this is the material where core and cell is there. So here this core is suppose silica, dielectric, and cell is of gold. So this can behave this particular nanostructure where core is silica, dielectric, cell is metal nanoparticle, and the medium is polyvinyl, that can work as a nano device. So in this way, here suppose then it is not necessarily that only one nanoparticle will be there. In an effective medium, lot more nanoparticles will be there. And as we know, for nanoparticle characterization, we can use transmission electron microscopy, scanning electron microscopy, then XRD, UV visible, everything, and according to the requirements and according to the our goal, we can use different. Just like suppose for targeted drug delivery, if we apply use for targeted drug delivery, then we will go for the absorption, scattering, all this. Suppose we want bioimaging, so for that we will go for the transmission and reflection coefficient. So our parameter will be different according to our application, and for different parameter, we will do different characterization, and accordingly we can use for this. So these are some different types of core cell structures. So metal core and different metal cell, that means we can use suppose silver nanoparticle as core and gold nanoparticle as cell. We can use gold and suppose silica, then silica and suppose the nylon, epoxy, all those things, then cadmium sulfide, zinc sulfide, then suppose like silver and cadmium sulfide, then suppose polyvinyl, then again resin, many things we can use. And even we can use some biofibers also, just like sugar cane, then this then neem juice, then many people use the potato, many people use the medicinal plants, the solution of the medicinal plants also. So these are the, as I have said, this can be used for core cell nanoparticles are used for biomedical applications, and for drug delivery, suppose we can use in this cell, we can use the, this is the scatterer, that means this is the nanomaterial. Here core and cell is there. Suppose our core is our suppose silica, and cell is suppose gold. When it is core and when it is cell, suppose this silica and gold is there, this silica gold core cell, and this is your matrix, suppose PBA matrix, and this is the perfectly matched layer. That means nothing can be infinite. If we are targeting some drugs, that must have also be a finite domain. So in this way, we can use the core cell nanoparticles for different applications. Just like as time is very limited, so I just want to show one. Suppose this is a structure. In this structure, this is a very famous structure, which is known as Yagiuda antenna, the antenna which earlierly in our TV it is used. Here this is a feed element and these are our directors and this is a reflector, and they have particular distance and this particular distance is maintained. Suppose like feed to director 0.3 lambda and feed to reflector is 0.25 lambda. In this way it is, and if we arrange nanomaterials or nanostructures in this way, then what will happen? That can be used for drug delivery application. Suppose here silica gold same material is used. The same silica gold material we are using as feed element, we are using as director, we are using as reflector. Just difference is what? Difference is the size band. For feed element, the size of the core is different. For reflector, it is different. For director, it is different. And the distance between the feed element and director and distance between the feed element and reflector is different. And according to that, we get the different directivity, different gain. And what we have observed that when it is one director, then also we are getting more directive. That means if we use silica gold nanomaterial, just like one example I am speaking, for targeted drug delivery application, that means it will directively, very directively, ultra directively, and ultra compactly, it can deliver the drugs, and it will not affect the other parts of our healthy cell. So our main target is what? Healthy cells should not be affected, and the cell which is having affected, that one should be cured. So in this way, the nanomaterials can be used for different applications, especially for the medical technology, and even in just like this. These are the characterization techniques, as I have said. XRD, we can do XRD, we can do HRTM, we can study the dielectric properties in this way. So what we can do, this can be this semiconductor material we can use for bioimaging. So nanomaterials with polymer matrix can be used for different applications. And since my time is, I think it's up till 3 p.m. So thank you so much. And ample opportunities are there for this polymer matrix based core cell nanoparticles, and it has very much applications in the medical domain. Since all the characterization techniques also speaks on that. And for accurate clinical imaging, just like we want to study or we want to image the breast cancer, we want to image the computer tomography techniques. There also what we can use for clinical imaging also, we can use the nanomaterials. And since we are using the broadband frequency range, suppose if we measure it from 500 megahertz to 20 gigahertz, then for a broader maximum domain, it can give us the informations. So thank you very much. I would like to acknowledge Department of Science and Technology, Government of India, Kharkhure Institute of Technology, Germany, IIT Kanpur, Guwahati University, CPET, CSTS, Guwahati, CRNN, Calcutta University for different laboratory facilities. And I would like to thank Dr. Mubarak for giving me this opportunity at this eve of the Women's Day. Thank you, Mubarak.
Okay, thank you so much, Dr. Jyotika, for your time, for dedicated for us to give an expertise in this forum. So I encourage all the participants can give your questions if you have. And Jyotika is happy to interact with all the participants what we have. And it's two more on electronics, so it's very maybe difficult to follow, but the application wise they could have understand much better based on your representations. So maybe wait.
Yes, that's why I have focused on the very preliminary things which are associated. That's why, because as I believe multidisciplinary people are here, so just an idea how the nanomaterials or any composite material can be used for the biomedicals. And the field is very prominent. So every people can go for that, because from different angles, different disciplines can join the research for that field.
Yeah, that's true. And I actually personally I have some question on the topic you have delivered. So regarding the core shell, we have some metal or any ions in the core and shell. So how it reacts actually, the shell, how it reacts with any of the ionic state or.
Actually, we use different materials. Suppose like we are using cadmium sulfide and zinc sulfide, or suppose silica and gold. Then they have different trapping and detrapping mechanisms. So at the interface, that polarization plays a very important role. And you can there are some of my papers are there for the relaxation mechanism. So that how at the interface, how the polarization will work, how that at the interface, how the relaxation behavior is using the trapping and detrapping at different frequency, it will behave. Actually for that only those things will come. Just like chemistry people will speak in terms of their view that the ions about the cations and anions, just like that. And from this angle, if we say from our medical application point of view, then this trapping and detrapping at what space or at what time the nanoparticles which are in assembly is trapped, and at what time it works as like pulse, you can say sometimes.
Yeah, actually, Dr. Asa Johnson had requested a question that how we have to choose specific polymer material as a medium for making of core shell nanomaterials.
Yes. We have to study first. Just like suppose we have PVA. Say example, we have water. Suppose we have potato juice. Then we want to use for different application. Then suppose preliminary we can go for the XRD. Suppose from the application point of view, we can go for the XRD. We are doing the XRD. Just like if we want the dielectric properties, suppose, then for water also, we can study for all the things. We have to do the study. Then we have to come to the point. Just like since time was not there, I had not shown. Just like I'm in that gold and nano antenna I have shown. There also I had did some comparisons. Just like whether PVA will be there, or whether stars will be there, or whether water will be there. So that we have to study and we have to compare. Just like in targeted drug delivery, what we want, that it should be moved to the NRI domain. So that means what? The wavelength should be red shifted. So if we compare those medium, then also which is having more red shift, we will opt for that. In that way, according to the application, we have to choose the medium.
Yes. So the Dr. Anjali also mentioned about the shelf life of the nanoparticles or effects in the functioning.
How nanoparticles affecting the functioning?
Shelf life of the particular nanoparticle, how it functions, whether it is effect on functioning or it not depend on its effect. Shelf life of.
Can you repeat again? Actually.
Yeah. Does the shelf life of the nanoparticle
Or
affect its
self-life? Yes, it will be different. Self-life means how normally what we say self-life. That means how we are improving our living quality, just like that. So depending on the earlier also, suppose like I need some mechanism. Suppose the nanoparticle functioning. Suppose just simple example, bioimaging. We want to use it for bioimaging. I need for a particular domain, suppose frequency range or wavelength range, you can say. Suppose I need it at lower frequency, radio frequency. That's my synthesizing mechanism, or how we will adopt anything, that will be different than for higher frequency. So indeed, in this way, we have to do when we want to improve the functioning, self-functioning. Then what we will do, we just like suppose someone wants to use it for bacterial properties, then definitely he or she will look for the aging, no? After 10 days, what will be its behavior, like that. So it's a process, and different applications it will be different. Just like bioimaging test, suppose I need it for lower frequency. At lower frequency, my particle size will be different, concentration will be different. And at higher frequencies, my concentration or the ratio for mixing the core and cell will be different. And definitely the pH factor also plays a very important role.
So, okay, so maybe one more question if you allow time. Actually, what will be the polymer metal complexes when biocomposite when we prepare? It surely affect that state. Either is a shift changes occurs, either it is a blue shift or a red shift occurs. So, what kind of change? I mean, what is the mechanism of changing its shift?
Shift, yes. Just like we have some particular bands. Just a minute. I am just.
Cap or only the cap?
Yes, I am just showing. Red shifting, how it is. Just like this one, suppose here, if we see, these are some shifting. So here we have the core cell materials are here. Suppose silica gold, gold silica, silver gold, gold silver, silica gold, like that. And what we have done, we have obtained the extinction, means absorption plus scattering. Then what we have found that depending on its behavior or depending on its size, just like if you go through this chart, you have found that, say, for example, gold core silver cell. In this core size is 66, cell size is 5. So this resonance wavelength we are getting at 730. And when we are using the core size 60 nanometer and cell size 11, we are getting 656. That means by tuning the core and cell size ratio, we can shift the resonance wavelength. I think I able to answer the question.
Yes. Actually, this is you skip that, so I maybe.
Actually, time was very limited, no? So I covered the main things. And here also, if you go through this, there's some size band, no? Same material, but its size are different. Suppose like 66 by 5 core cell. Then what we have find that for the 66 by 5, we are getting the maximum shift. So the total is same, 71 nanometer, but the cell and core size are different. So different size when we are taking, then we are getting the different resonance.
Okay. Thank you, Doctor. Maybe the further question, if anyone have, then they will be contact you if you're able to answer these questions. And very much, I mean, I can share.
Thank you very much. Thank you very much.
Yes. Thank you. Yes. Actually, your
thank you so much.
breaks. Yeah. Okay. Thank you so much, Dr. Jyotika. The next speaker also ready now. Maybe you can allow time to speak. If you wanted to say anything, you can.
Yes, if my time permits, if my time permits, I will also be there for that. Thank you so much.
Thank you. Okay. Yes. So next speaker with us is Dr. Ingo Mbak Medellin Nina. Actually, she's a senior lecturer from the University of Douala, Cameroon. Nina, are you available, right?
Good afternoon. Good afternoon, Mubarak.
Yeah, Mr. Barak, I just introduced. Okay, Dr. Engo Mbak Madeline Nina Lau is a lecturer at the Department of Biochemistry, and she obtained her doctorate in the Biochemistry department. She specialized area in the drug discovery from the biomolecules, and she have received many awards and grants from various funding sources from the French Society of Medical Mycology, and she got many travel grants, and through that, and also from the scholarship of CV Roman scholarship for the Indian government for the young African researchers. Through that program, she visited India for 6 months, where to my lab where I have been associated. So we had a very good time when she stayed in India, and she's now teaching the biochemistry and medical microbiology in Cameroon, and also researching on especially the candidiasis of African population. And she's an excellent. She got an excellent award from the President of Republic of Cameroon, and many other noteworthy contributions. So with that, I like to welcome Nina for giving her lecture now. Nina?
Yes.
I think your camera is somewhat focusing ceiling. Can you keep down? Yeah, that's.
Can you see me, please? Are you able to see me, please?
Yeah.
Okay. So good afternoon to everybody. Good afternoon to Dr. Mubarak. Good afternoon to all the organizing committee, and also to all the university, the administrative members of the university also. So I would like to take the opportunity to present a special topic about the antimicrobials focused on the biotechnology. But before continuing, I would like to first, like a remembrance, I would like to say, to wish a happy Women's Week for the International Journal of the Women, which was yesterday. So it is, I think it's a great moment for women scientists to share their knowledge, to share their experience, their scientific experience to all the communities, to other women and to other scientists also. So thank you again for that opportunity. So I'm trying to share my slides. Okay. Okay. Please, I hope that it is okay.
Yes,
so,
can you go to the presentation mode, Nina?
Please.
Presentation mode. Actually, it is shared, but can you give a F5 or presentation mode?
Yes, I'm sharing the PowerPoint. I don't know if you have received the slide.
I seen the slide.
Yes. Okay, so I would like to start the presentation. Is it okay? Are you ready?
Expect we seeing it's not in the presentation mode. Can you press that fourth icon? There is a first icon. Can you go to the fourth icon at the bottom? You can find the 65 next to the 65. There is an icon. Yeah. Maybe you can share the entire screen. Can we stop share and then we can do it back? Then it will becomes to share entire screen. Then you can go there.
Okay. It's okay. I don't know if it's okay.
Yeah. Okay, can you please proceed? Maybe it is visible only. However, it's fine. We can proceed.
Okay. So I'm trying to present slides. I hope that everybody can see the slides. So I will present a topic concerning antimicrobials from natural sources against pathogenic fungi in the framework of the International Conclave on Women Scientists in Science and Technology, with a special reference to biotechnology. So why have I choose this particular topic? Because we have observed that antifungals or fungal infections have a great impact as opportunistic disease in people which are immunocompromised or immunodepressed, like HIV, cancer patients. And this disease could cause a lot of death in the population or a lot of uncomfortable situations for these patients. And we can also see that those infections, those fungals or these fungal infections could have an impact on the crops productions in the field of agriculture or agronomy, and could have also impact in the field of the production of foods like fish and even bovines also. So it is an important subject because there are a lot of problems in the field of antifungal drugs. Okay, so I will proceed with the next. Okay, so as the application of antimicrobials from natural sources against. Yes.
Are you changing slides?
Yes.
But it's not reflected, right? We are seeing only the front slide. Can you stop screen and then can you share again?
Okay.
With the yeah, we can share entire screen, so it will be possible. Don't share the slides, only entire screen, then you can able to.
Okay. I'm opening the sharing
option where there will be plenty of icon will come, and go to this major slide.
Yes, I'm going major slides.
Then you share.
Is it okay?
Yes, now it is fine. You can proceed now. Thank you.
Okay. Do you see the?
Yeah, this is second slide, I sense.
Second slide. Okay.
Yeah,
do you see the third slide?
Yeah.
Okay, it's fine. Yes. So I'm sorry for that inconvenience. We will continue. Okay. So as I was saying earlier, I'm presenting a topic on antimicrobials from natural sources against pathogenic fungi. So I've already given the importance of the topic. So why have I choose, or we have choose these important subjects about antifungals? Because of their impact on human health, and the impact also about the production, crop productions, and also bovines and food production in the level of aquacultures. Okay, so now we like to make or to simply explain the link between biotechnology. So as a simple definition, biotechnology is the use of biology for industrial purposes. So it means that we can use microorganisms, we can use plants for many industrial purposes. And as industrial applications, we can have pharmaceutical industry, or as industry, available industry. We can have a pharmaceutical industry. So we can have food industries. We can also have petrol industry. And all those, or these particular industries, need biotechnology. So biotechnology from microorganisms, either from plants, nor for any superior organisms we have. So as we say that the biotechnology is the use of biology for industrial purpose, or the production of industrial products, it means that we need to have a natural sources or a biosources. So what could represent those natural biosources? So we have living microorganisms, for example, included bacteria, fungi, and viruses, and many other microorganisms also, which could be a natural source for those kind of compounds and even processes which can save for industrial development or industrial products for development. We also have living organisms, including superior organisms like plants and animals. So we have the main sources, the main natural sources for biotechnology according the particular topics we have, according the topic of antifungals against pathogenic fungi. So we have these living microorganisms and living superior organisms. Okay, so as definition, we have discussed or we have talked earlier about antifungals, about the drugs against fungi. So we can simply say that antifungals are drugs used to treat fungal infections caused by a fungi. So the infections will be caused against the human health. The infection also could be developed on crops. And as infected crops, we can see different disease of those kind of fungal infections at the level of the crops. So we have anthracnose, we have botrytis rots, we have downy mildew, we have fusarium rots, we also have powdery mildew, we have rust, we have rhizostonia rots, sclerotinia rots, and sclerotium rots. So all those fungal infections would affect crops and reduce the production at the level of agronomy or at the level of agricultural items. So it is really important to control or to manage those fungal infections if you want to ensure the economic development and the food security also in particular region in the world. So it is an important topic on that particular level. And here we can see different pictures about infected crops which have been attacked by particular fungi, pathogenic fungi. So here we can see the picture of a mildew affecting potatoes. So we can see how the potatoes is affected. We can see the characteristic of unaffected potatoes. So, and then we also have a maize which presented destruction zones because of that mildew. And we also have a fusarium roots of a soybean which have been destroyed by the pathogenic fungi. At that level, the plant is unable anymore to grow and to produce the fruits or the seeds. So that's why it is important. So at that level, we can see that the production will be really decreased, and we have a direct impact on the economy and food security also. Then we have another, how can I say it? On another hand, we have the impact of fungal infections on the human health. So here, human health or human beings could be infected by fungi, beasts and fish also. And at that particular level, we have opportunities. There are opportunistic fungal infections. We have said that why opportunistic? Because they can develop, these fungi can develop themselves or itself just because the organism of the host is immunocompromised or immunodepressed, or the immunity have been suppressed because of particular kind of therapy, or for example, for cancer people or cancer patients, or because of a particular physiology, maybe for diabetic persons, which have an immunity which is really low already. So those opportunities develop themselves like this, because of a lack of the immunity system at a certain level. So we have as an example, or as examples, we have candidiasis. We also have aspergillosis, we have cryptococcosis, We have dermatosis and many other kinds also of fungal infections on animals, means human beings, beasts, and fish. And we also have to notice that those infections could be systemic also. We call it systemic mycosis or systemic fungal infections. So they can spread themselves or spread around the whole body. Means that they could affect different type of organs, or maybe the whole organs inside the organisms. Okay, they can also affect the blood and cause septicemia. It is also a kind of systemic infections, and people could die because of those kind of infections. And the percentage of dying people is increasing. Okay, on that picture, we can see different images of the representation of fungal disease on human, beast, and fish too. So on the first picture, we can see at the level of lung, at that particular level of lung, we can see that cells could be affected by fungal infection and cause pneumonia. And we can also see here edema, phytosis. We can see the clinical manifestation of a mycosis. So we can see the inflammations at the level of the skin. We can also see that the person can suffer on some irritated zone. So it also can be really painful and create uncomfortable situations. And we can see that also in the picture that the person is an old person. So elderly people are really exposed also to that disease, or to this particular disease as opportunistic fungal infections. And on the third pictures, we can see that the fungal infection, we can see some zones or some surfaces on the fish skin or on the fish flesh. We can see the development of some affection or some area which have been infected or caused by the pathogenic fungi. So this infection would create the presence of toxins inside the food or inside the fish, which are supposed to be eaten by the population. So it could also cause a source of poison. So it can also be the poisoning source. So when food or fish or animals which are focused for use, or which are used in common daily food system in many places, so you can be contaminated by some toxins which are present inside the fish. So we know that we have natural inner toxins inside some animals, but here also some microorganisms like bacteria, but here we are talking about fungi. Even fungi could also lead to the development of some toxins which can be a poisoning inside the human life. Also at the level of the ecological system, so it could cause the destruction of a whole species. So it is really important to manage those kind of infections. Okay, so in the way to control those fungal infections which have a considerable impact on the health, also a considerable impact on the production, the crops production, and directly on the economy and food security of many regions worldwide. We have developed, or a kind of antifungals have been developed. We have here 4 groups of antifungals which have been developed. We have polyen. It is not like particular drugs, but it's just like a group or groups of antifungals. It means that they have been grouped or they have been assembled just because of their targets. So all these 5, each 5 groups here have particular targets. So inside polyens, we have, we know about the nystatin and amphotericin B, which are drugs belonging to that group of antifungals. So we have polyen, we have azole, we have echinocandin, we have pyrimidine, and allylamine. So all these 5 group of antifungals have particular targets, cellular targets on the pathogenic fungus. Okay, so here we can see that the quantity of those antifungals are really small. So the quantity is not sufficient to face the problem of resistance, antimicrobial resistance. We know that we have emerging pathogenic fungi. We also have those who have developed a lot of resistance as biofilm, as efflux form also. So then we have few kind of we have a few diversity of antifungals. So there is a need to develop the research or to go deeply inside research to find or to discover new antifungals drugs with different targets and new targets also. Okay, so on these slides, we will see different mode of actions of these particular antifungal drugs. So we have pathogenic, we have the representation of a pathogenic fungi. Yes, it's a common one. So when we use polyen, for example, to treat a simple candidiasis or to treat or to inhibit Candida albicans, for example, or Cryptococcus neoformans. So when we use polyen, which are highlighted by nystatin and amphotericin B, so we see that polyen could target ergosterol by a complexation system. So polyen will enter inside the cell through the membrane and will complex ergosterol, which is present at the level of the membrane. So when polyen will target ergosterol, which is present at the level of the membrane, it will cause a lack of equilibrium at the level of the membrane. And then the membrane will lose its equilibrium, and then all the cells will be destroyed because of a leakage. So everything will be, the cell, the inner components of the cell will be drawn outside of the cell because of the leakage. So the membrane will be destroyed or disrupted. Okay, and also we have a problem of toxicity of these particular groups of drugs. Why? Because we also know that inside normal human cells, we have ergosterol inside our inner membrane. So we have ergosterol, and this polyen also could target the same molecule. So when the targets, we have a cholesterol, yes, which is not really different of ergosterol. So it could also target it and create some toxicity and destroy some cells. And we also have the problem of the low solubility in water. So we have polyen are really, they do not have a good solubility inside water. So that means we have the problem of nephrotoxicity. Okay, and then we also have azoles. So this group of antifungals with precisely targets an enzyme. So the enzyme is highlighted at the level of the ergosterol synthesis pathway. So they will target the pathway, the enzymatic pathway of ergosterol synthesis. Okay, and here we have the alpha demethylase. So they will target alpha demethylase. So as an example of azole, we have fluconazole, we have miconazole, and many others also, which will target or which will inhibit that enzyme. And then the inhibition of that enzyme will automatically decrease the production of ergosterol inside the cells, and which also we have a problem of the membrane. So the membrane will not be renewed because of the lack of ergosterol, which is an important constituent or component of the cell membrane. Actually, we also have allylamine. Allylamine also will target a particular enzyme, and that enzyme is squalene epoxidase. This squalene epoxidase will also act at the level of the ergosterol synthesis pathway, and will block or we stop the production of ergosterol inside the cells. and then it will create the inhibition of the cell. Fourthly, we have also mitosis inhibition, which is glycerofulvin. And this glycerofulvin will block the division of the cell at the level of metaphase by blocking or by stopping the polymerization of microtubules, which is an important item in the cell divisions. And we also have DNA blockers, like DNA inhibitors. So this kind of antifungals, we are calling them pyrimidines. So they will block or they will stop or inhibit DNA, the synthesis of DNA, and lead to the substitution for uracil. And finally, we have echinocandin, which particularly inhibits the synthesis of the cell wall. So if we want to do a little sentence, we can see that the main targets for antifungal drugs are the membrane. We have membrane, we have cell wall, we also have ADN at the level of nucleus, yes. So we have ADN and these two others targets. So we can see that the targets of antifungals are not yet really diversified. So there is a great need in the research for new antifungals with different mode of actions or with different targets. Yes, because the research on that level is really few, but the impact on the health and also on the crop production is really increasing. So actually, we sometimes propose the combination therapy. What is the importance of the combination therapy? So we can combine two products, we can combine two compounds to have to increase, yes, the activity. Why the activity is increased? Because these compounds could target different cellular targets at the same time in the aim to face the problem of resistance. Okay, so here we have seen that there are many alternatives. We have seen the alternative of microorganisms, and we also know that we have plants, which could be an alternative for drugs discovery. And here we focus our attention on microorganisms. Okay, so in biotechnology, we have seen that we can use microorganisms to develop or the function, the growth or the functions of microorganisms to develop industrial products. Yes, in that particular level, we can use microorganisms to develop industrial pharmaceutical products. So industrial products in the framework of pharmacy. Okay, so how living organisms can help us to have these antifungals. So we have taken the example of penicillium. Yes, and on that picture, we can see some development of a particular microorganism, which is a penicillium species with green-blue mycelia. So we have the development of the mycelia with particular characteristics. Then we can see all the physical characteristics of this particular mycelium or mycelia on the medium inside the petri dish. Okay, at the microscopic level, so when we see the microscopy of the microorganism, we can see a function of like a little tree or a little flower. Yes, it's really beautiful. That's also how can I say, the magic of microbiology. So we can see a little conidia. We can see that little cycle, that little round items, round structures at the ends of the main structures. They have conidia, yes. And then after the conidia, we have phyalids, we have matrule, and we have the conidiophore, the ramificated conidiophore coming from the main one, which is well behind. So we have the microscopic image or the microscopic picture of an example of a fungi or a microorganism which could produce some industrial interest products. And we will see during the next part of the presentation how these microorganisms could be saved as a source of biotechnology development. Okay, so inside these cells, we have a lot of components. We have a lot of organoids, yes, inside the structures of these microorganisms. And as we have said, that penicillium that we have seen, the microscopic characteristic and also the physical characteristic, they can produce some bioactive compounds. And these bioactive compounds, among these bioactive compounds they could produce, we have penicillin, which is an antibacterial compound, and we also have a glycerofluorine, which is an antifungal compound. So during the presentation of antifungals, yes, we have seen glycerofluorine as an example of antifungal drugs. So these antifungal drugs have been isolated from a particular microorganism that we call penicillium. Okay, so glycerofluorine is an antifungal medication used to treat a number of types of dermatophytosis. These include fungal infections, and it can be taken orally at this way of administration. We can take it orally. And glycerofluorine also was discovered in 1939 from a soil fungus that we call Penicillium glycerofulvum. That is the right species. We have Penicillium glycerofulvum. There are a lot of species of Penicillium, and there we have the particular, the specific one which can produce penicillin in a great quantity. And also here we have the molecular structure, the chemical structure of the compound. Okay, so we have to know that all these components or all these bioactive compounds which are produced by microorganisms are called secondary metabolites. And they are not the sole metabolites produced by these microorganisms. We have also primary metabolites which could be produced by these microorganisms. But here we are focused our attention on secondary metabolites. Why? Because they are bioactive ones. But we could also have some bioactive compounds in the field of primary metabolites. But in the grid representation, we have secondary metabolites, which include a great quantity of bioactive compounds. So how could we extract, or how could we have compounds like metabolites or secondary metabolites, primary or secondary metabolites from a particular microorganism? So how could we obtain those compounds? It is simple, it's just through fermentations. So we take the microorganisms and we proceed to fermentation, and we can extract a lot of microorganisms. So fermentation is what? It's simply explained by a microorganisms which is in contact with a source of substrates, with a substrate which can be a carbon source, which can be a nitrogen source or a minerals. So when the microorganism is in contact with a particular substrate, it will start the digestion of these substrates, and it will produce, or it will lead to the productions of many types of compounds or metabolites. And then during the production of those metabolites, all this process is called fermentation. So we have a substrate which will be digesting in many types of other components by the production and the consumption of the energy by the microorganisms. Okay, so first we have, as we have said, the microorganisms need to grow. So we will grow by digesting substrates. We have said that we have carbon source, we have nitrogen and minerals. So after digesting all these substrates, the quantity of the microorganism will increase. So we will have the biomass increase. It means that the quantity of the microorganism, or the number of the cells will increase, will automatically or dramatically increase. And because of the increase of that microorganism, we also have a great percentage, a great yield of those metabolites. Here we do not have a particular metabolites, but we have a great quantity of metabolites. And inside these metabolites, according to the site of the production of the metabolites by the microorganisms, we can have endocellulars. It means that inside the cell, mean that the penicillium or the bacteria or any else microorganism will produce metabolites inside this, inside the cytoplasm or inside the cells. So those metabolites are called endocellular according to the site of the production. And then we have also exocellulars and metabolites because they will be produced outside of the cells. So in the environment of the growing environment of the microorganisms. Now, concerning the type of the metabolites, we have primary metabolites and secondary metabolites. So what is primary metabolites? These are metabolites which are a little bit produced. They are intermediate metabolites, little bit, they are produced for the growth of the cells. We can have alcohol, we can have proteins, we can have lipid and many other. And then we have secondary metabolites. We are produced laterally by the microorganisms, and they are generally used for the defense purposes, for the communications between cells, like signalizations and many other. Okay, so here, on that slide, we can see the industrial process of the production of the fermentation of penicillium for the production, for example, for penicillins, or it could be also the production of another type of microorganisms. It depends on the compounds you are targeting, or the secondary metabolites you are targeting. So here we can see a batch fermenter. So inside that batch fermenter, we have the presence of sugars, the presence of substrates, different type of substrates, and the conditions also are well controlled. So the main information is here is the presence of sugar, the presence of the substrates. So all these substrates, even nitrogen salts, they are mixed and put in contact with the penicillium or with the microorganisms that we need the production of secondary metabolites. And this generally called starter culture. So after some days, the culture media will be replaced. Why? Because the substrates will be, we need to replace the substrate source because the microorganism has been already digested all the available substrates inside the medium. And we suppose that the secondary metabolites have been already produced. And then we will change the medium in the aim to increase the production of new secondary metabolites, otherwise in the aim to increase the fermentation process. Okay, so after the fermentation process, we have another process which is here. We have the extraction process, the secondary or the metabolite extractions. If you want to extract primary metabolites, you can also perform an extraction focus on primary metabolite extraction, or you can also develop a process for secondary metabolite extractions. So after the fermentation, the filtrate will be taken, and then all the steps will be performed up to obtain these new antibiotics or enzymes, what we are looking for. This is an example of industrial fermentation for a particular microorganism, as we have said, Penicillium genus. And here we also have biochemical processes of fermentation. So what is done at the level of biochemistry? Here we have an example. There we have an example of industrial process. Then we have an example of biochemical process. So if we take the example of glucose, we can see that we have a lot of primary metabolites which are produced. We have pyruvate, we have acetyl-CoA, and you have a lot of these compounds which are produced, which we save for the growth of the microorganisms. In general, it is produced during a particular phase of growth of the microorganisms. And then later, this is produced at the early stage of the growth of the microorganisms. And then laterally, we will see the production of our secondary metabolites, our bioactive compounds that we are looking for. So here at the beginning, at the end, we can see penicillin. If we are looking for a particular kind of compounds, so we have secondary metabolites at that particular level. That is just an example for a biochemical process of fermentation. Okay, so after the fermentation, we said that we need to perform the extraction process. So after performing the fermentations, we need to do the extractions. But we have said that during the fermentation, we have proteins, we have alcohol, and many other compounds which could be produced inside the cells or outside of the cells. And we have said that we have endocellular metabolites and exocellular metabolites that are represented here on the picture. And after it, we need to proceed with the extractions. So for the extraction of those metabolites, if we have endocellular, we need to proceed first with ELYSIS. But if not, we can simply continue the extraction without doing any lysis, cell lysis. But it is really important to perform always cell lysis. Why? Because we want to increase the extraction need, and we never know where, when you are doing a screening, we never know where the targeting compounds could be found in the field of drug discovery, because sometimes we cannot know if it is which proteins or which type of secondary metabolite which could highlight the activity or exhibit the activity. So it is better every time to perform the lysis of the cells in the aim to obtain the whole or the total quantity of the metabolites produced by the microorganism. Then we can do targeted purifications, then to purify and isolate pure compounds. Okay, so at that level, we can do then centrifugations. After the centrifugation, we can do a simple filtration. And at that level, we will take the filtrate. After the centrifugation, we will take the filtrate, do an evaporation, a precipitation up to the drying process, and we obtain our extract. And yeah, that extract we have obtained is it's not always pure, as we have said. So after obtaining that extract, we can perform another type or other types of chemical separation or chemical extraction. Again, we can perform chromatography, HPLC chromatography, preparative or separative chromatography, particularly separative chromatography. And with those extracts, we can couple that chromatography techniques with spectrometry to identify the precise identity or the precise name of that compounds. Okay.
Dr. Nina?
Yes?
You have 5 minutes more.
5 minutes, okay.
Yeah. Can you make it fast, please?
Oh, okay, thank you. Okay, we go to the next slide. Okay, actually, we can see on that presentation, on that slide, we can see the two phases or the two steps of the two steps of the growth of the microorganism growth. We have seen that we have the first phase, which is a tropo phase or the exponential growth phase of the microorganisms. And during this exponential phase, the exponential growth phase or tropo phase, we have the production of organic acid, the production of alcohol, polysaccharides, and lipids also. And also we have the ideal phase, which is the stationary phase. At that level, the microorganism will not grow fastly anymore. So it's a constant phase. And during that particular phase, the microorganism will produce secondary metabolites, which are unrelated to cell growth. So they are really used for toxins production. They are really used for toxin production in the hem to allow the defense of the microorganisms, the signalizations, and the communications also with all the surrounding microorganism around it. Okay. And here on the slide, we can see that we have a lot of example of secondary metabolites which have been isolated from pathogeny, from producing fungus, for many fungi also and the applications. And we can see that we have an immunosuppressant, we have pigments too, we have plant growth regulator to improve or to increase the crops productions. We have antifungals, as we have seen. We have seen early. We have glycerofulvin coming from Penicillium glycerofulvon. We have cholesterol-lowering drugs. We have another immunosuppressants. We have a lot of antibiotics like penicillins. We have seen the example. We have the main compound Taxol, which is an anti-cancer drug today, which have been isolated from microorganisms. So that's the importance of the biotechnology using microorganisms, secondary metabolites productions. And also it is important to mention that it is just secondary metabolite productions. It's just an aspect of biotechnology, but here we can also stimulate the genes production of these secondary metabolites. So we can have a microorganism transfer inside a microorganism which can easily grow fastly and then increase its potential or ability to produce a particular secondary metabolite, or transfer inside it a gene which can produce a particular secondary metabolite and have our metabolite. We can also use a particular process inside the biochemical processes of the microorganism to use it also as a mimic system in the field of pharmaceutical sciences. Okay, here we also have the example of nystatin and amphotericin B, which have been produced not from a fungi, but from a bacteria. That bacteria is Streptomyces nodules. And the same also could be produced from another fungus, but here the production is low. That's why I've said that you can activate a gene production of a particular secondary metabolite inside another pathogen, maybe which can grow fastly than the other one. Explore these abilities also in biotechnology. We can also have nystatin, which have been isolated from Streptomyces nodules. And here we have some emblematic, we have an emblematic figure in the field of antifungal drugs discovery from microorganism isolation. So here I just focus my attention on the field of antifungal drugs discovery. We have many important women scientists around the world, and here they are the pioneer in the field of antifungals discovery, drugs discovery. So we have Rachel Fuller-Brown and Elizabeth Lee Hazen, which who have discovered antifungal agent, which is nystatin in 1950. Rachel Fuller-Brown. Okay, Rachel Fuller was a chemist, and the other one a microbiologist. References from the references from that presentation have been taken from online scientific review, from the literature available on Elsevier, Springer, and other one. So we cannot conclude, we cannot close our presentation without thanking the Crescent School of Life Science, professors and women scientists involved in the conference, conference organizers, and all the participants. Thank you.
Thank you very much, Dr. Nina, for presentation all the way from the Cameroon. Really good to see you after a long time, and then I hope the participants have enjoyed your detailed information on the antimicrobial system, the fungi. So the participant, if you have any where you wanted to talk, then you can able to. films. But simply say back there is some. Since it's not full screen, right? It's full screen.
Hello, Prof. Mubarak.
Yeah.
Is it okay? Do you have any questions concerning?
Yeah. That's why actually I'm looking for the chat box.
Okay.
Yes, actually the question from Dr. Sharika Bosle. Please name any advances or novel antifungal or newly emerging or the newly emerging pathogen. Any novel antifungal compound?
Yes. Okay. Thank you for the questions. We have many innovative antifungal compounds which are still on the research process. So they are not yet conventional drugs, antifungal drugs, which have been put on the market. But yeah, the research are still ongoing. That's why I say that the quantity of antifungal drugs available is really small. So there are some compounds which are still on the ongoing research process to be confirmed before put the antifungal drugs on the market. But there are many new or promising sources of microorganisms or antifungal drugs. We have, for example, marine compounds, which could be also a promising source of antifungal drugs. But yet we just have 5 groups of these antifungal drugs, and we need to search new antifungals. Maybe it will be one woman or one person to discover the next new antifungal drugs. Okay, I don't know if I
hope they have. Is there any question? Yes, maybe thanks about it. I think if there are any question, then I will forward you, Nina, then you can able to answer for the question. Thank you for joining and then your presentation. That's Nina?
Thank you to Prof.
Yeah, thank you.
As a pleasure. Thank you.
Yes. Yeah, thank you so much. So now we all come to this next part of our presentation is tribute human scientists. So that's what I'm gonna present now. Today, and also again presenting top 5 or top 10 slogan that you have sent during the registration. So with this, I take privilege and very honored to give a tribute to the human scientists in India at various sectors. So as you all know that the beginning of the women who excel in the different sectors, right, from the finance, biotech, and the IT field, you know, the popular figure in India, they're excelling it, but and also the inflow scientists are working on different aspects. And I'd like to here show you the important as a person who contributed for especially for the biotechnology, right, for the cytogenetics and botanists, biochemists, chemists. There are so many Indian scientists who have been they contributed a lot for this development of our nation. So among which I have to mention one that is Dr. Janaki Ammal. So she used to call in a sugarcane queen because she created several yielding hybrids of variety of sugarcane. And this is she is known for her field of cytogenetics and the phytogeography, and also used to collect many novel plants which have the economical value in the rainforest of Kerala. And she is honored with Padmashri in the year of 1977, and with her name, the Department of Environment and Forestry initiated a national award for the taxonomy with her name. It's called the Janaki Ammal Taxonomist Award. Still, this award is conferring to the women scientists who are working in the botany or the taxonomy field. They can avail this award. And this is an Indian pediatrician and also clinical scientist, that is Soumya Swaminathan. And actually, she is from the Chennai, and then the daughter of Swaminathan, Emma Swaminathan, and she is serving now as an chief scientist and also deputy director of WHO. So these are the prides of the women and scientists in India who were contributors for the same. And now we have come to the point that is top 5 selected slogans from the different categories: scientists, student, and also scholars. So with this, I like to congratulate Subarwa Sagu, Nashat Nazir, Varlakshmi, Jasmine, Juliet Richard, and Tajul Westin from various institutes. They given the slogan as I mentioned, because it's very big, we cannot I can pronounce many things, but I can read one. Nashat Nazir, his woman to the real architect of the society. So that's what our slogan. And this is under the category of research scholars. So at the various institutions, there are plenty. Which one I read? All big. Okay, from Kanaham Nachiappan from Vellore Institute of Technology, is everything revolves around a woman, be it home or science. So that's the slogan he pointed out. And the name I can read: Rashmi Kondi, Raya Bhattacharya, Lubna Siddiq, Sumaila Banu, Jay Kumari, Kanaham Nachiappan, Ashwini John, Tripathi Raghavendra, Durhana Yasin, Kailai Naik. So all are the selected research scholar category. And next is student category. So Tasmia Fatima, Nazmin Akhtar, Aparna, Mohammed Sohail Apasi, Sudha, Trisha, Akansha Srivastava, Tashmia Fatima. Oh, there is 2 entries. Nazin Akhtar, Aisha Hussain, Ikra Yunus, Daksha Cruz. So these are the person from the student category, and one is from Pakistan, Ikra Yunus. So that's a very bigger quote. However, I can read for their maybe contribution in spite of many aspects. So Ikra Yunus stated the slogan as a very big slogan maybe. Women today are unstoppable, whether it is engineering, medicine, media, defense, sports, finance, technology, or art. It's difficult to find a field where they have not said their foothold. Along with the excelling in the professional arena, it's not forget how a woman also balances her personal life and nurtures a family. A progressive woman is an inspiration for the society. A charming woman doesn't follow the crowd. She is herself. So with the quote, they ended up. So congratulations for all the categories who have awarded. So I think totally we received 400 slogans so far, and Dr. Rihana from Jamah Muhammad College, and then Dr. Farida Begum have acted as a referee to choose in the different categories based on their merit. And the selected slogan creators will be honored with a surprising gift during the valediction that is tomorrow. So stay tuned for the same. And now it's time for the feedback. You can note this link. And also I post the link in the chat box. To the chat, the feedback link is mandatory for receiving your certificate. So the end of the day too. Thank you so much for joining. Have a nice day.